All right, welcome to Friday. Get used to these 4-day weeks, but I posted a number of things yesterday online because our first exam is a week from today. So honeymoon is almost over. It will include up through chapter 3. So I'm hoping, I'm pretty confident that I can get through the end of chapter 3 on Monday. If I have to bleed a little bit into Wednesday, I'll try not to, but I don't think we're going to get to that next chapter on the eukaryotes. So I made that eukaryotic chapter homework not due until the next exam. So just your homework up through that chapter 3, these cell structures that we've been talking about. So this is a little bit less material covered on this exam than what will be on the other ones. I still think there's plenty of things I can ask you about, but just to kind of ease into the semester just a little bit. I have the exam set up as being 46 multiple choice questions. So I think 46 multiple choice questions in 50 minutes is reasonable. Might even have a few more on the next exams. I don't like to rush you too much. This exam next Friday, I could give you an extra 10 minutes or so in class, but whenever we have exams on Monday and Wednesday, I have to go straight to class after this, so I'm not able to do that. But this one I can give, maybe a little freedom there. But it is all multiple choice. And I think we should talk about how to study a little bit. I know this is not your first college class, but I know when I studied for classes like this that were really content heavy in college, I tended to just read over my notes. That's how I studied. And sometimes I would get to the exam and I would realize things that I thought I knew, I didn't really know. In other words, I was familiar with things, but I didn't really know them. And I see that happen a lot in this course. So to prevent that, you have to test yourself ahead of time to see if you know it. All right, so say you're studying the cell wall. Okay, don't just read through your notes. Cover up part of your notes and say, okay, what do I know about the gram-positive cell wall? If you can't come up with anything, then you didn't really know it. All right, so, oh yeah, peptidoglycan. Oh, it has something to do with that. Okay, so you were familiar with it, but okay, what is that structure? So, practice kind of asking yourself what kind of questions that you think I might ask you. And for some idea of that, you can look at the learning objectives. All right, so everything I ask you is going to be something that we talked about in class, or there occasionally could be sort of an application question, that you had to know something that we talked about in class to apply it to something else. Some of these homework questions, like there's a question that I don't like that I'll get rid of next time, that asked about who we think the true inventor of the compound microscope. Well, in class, we said that Robert Hooke was the inventor of the compound microscope, and Leeuwenhoek was the inventor of the simple microscope. Well, in the book, they kind of dicker about, well, we think maybe it actually might be this other person, and that's sort of interesting because it kind of helps us understand how science works. Sometimes, we don't have all the facts and we give credit for something, but study your notes, study what we talk about in class, use your book as reference. I mean, people learn different ways, obviously. Some people learn better just by reading something. The homework is supposed to be reinforcement, maybe to get you into that text a little bit if you haven't been already. So questions about that, what the next week is looking like. Okay. So we'll continue with our cell structures here. We're talking about prokaryotic cells here, in particular bacteria.
Earlier, said your mic was muted or something.
Yes, thank you. I think that should be okay, because actually I have to do a little bit of muting so I don't get too much feedback. So let me just double check, but I think we're okay. thanks. Okay, so we were working through our cell structures from the outside in. So we talked about calyx and flagella, and getting to the cell wall. So here's a good example here, how I'm talking about how the cell wall grows. Okay, that's not mentioned in the book. It's a little bit extra, so you're going to want to pay attention again to what we talk about in class. And I felt like at the end, I kind of hurry a little bit right at the end, but that peptidoglycan structure, which remember is unique to bacteria. Any bacteria that has a cell wall is going to have peptidoglycan in the cell wall. And archaea or eukarya, if they have cell walls, does not have peptidoglycan in it. So that peptidoglycan, in order to grow, it has to be kind of chopped up and new subunits added in so that it can grow. And we talked about the action of autolysins, which break those bonds between the sugars as well as the amino acid cross bridges, and the action of transpeptidases, TP in this picture here, that binds up those amino acids. So what I didn't get to showing you, let's see how long this takes to come up, but this is just like a 7-second animation of what happens when a bacterial cell is in the presence of penicillin or another antibiotic like penicillin. It binds to that transpeptidase. And so essentially those amino acid cross bridges can't reform, and that impacts the integrity of the cell wall. And when the cell wall is not whole, then it's going to be susceptible to osmotic pressures. So this is time-lapse photography and full sound effects added, but essentially the cell lyses if the cell wall is not intact if it's in a hypotonic environment, which it often would be. So an application that we'll talk much more about antibiotics as we go forward. All right, so there's really two main kinds of cell walls in bacteria. And we call those the gram-positive cell wall and the gram-negative cell wall. And we mentioned this earlier because we said this is kind of the first question we ask when we're trying to identify an organism. Is it gram-positive or is it gram-negative? So let's look at the differences there. Gram-positive cell walls are composed primarily of peptidoglycan. Okay, so lots peptidoglycan. About somewhere between, it varies between cells, but somewhere between 40 to 80% of the cell wall dry weight is composed of peptidoglycan. Okay, so a nice mnemonic there. Gram-positives, thick, lots amount of peptidoglycan. And as we'll see for the gram-negatives, okay, they have a very thin amount of peptidoglycan. So that's kind of a nice little mnemonic. What else is in there? Gram-positive also have teichoic and lipoteichoic acids. Okay, these are actually sugars that increase the rigidity of that cell wall. We think there might be some other functions to those sugars, maybe some attachment functions. But the main thing here is that we only see those in gram-positive cell walls. So if we're gonna, it's always nice to have a picture. I like the pictures from our old textbook a little bit better, so I kept those for the most part. But if you look at a gram-positive cell wall, all that pink stuff that kind of looks like a hamburger patty or something like that, that's peptidoglycan. And then we've got our kind of anchored within that teichoic acid. The lipoteichoic acid is the same, basically same thing, only it's anchored down here in the plasma membrane. So it's really not a particularly complicated structure in terms of the gram positives. Okay. One other thing that I forgot to mention that I wanted to up here. Okay, if you're doing a gram stain, they are going to stain purple. And I know I didn't really talk about the steps of that last time, but essentially the first dye that you use in that gram staining procedure, because there's so much peptidoglycan, it holds on to that dye and it doesn't get decolorized very quickly. So that's reasonable to know that it's going to be purple. All right, contrast that to the gram-negative cell wall, which is a little bit more complicated. Like I said, much less peptidoglycan, maybe only about 10% of the cell wall by dry weight is going to be peptidoglycan, so thin layer. And because there's so little peptidoglycan, that first dye that we use when we're gram staining gets decolorized really easily. And so we have to add a second dye so that we can see it. So it stains that red of that second dye that we use. What's really unique about the gram-negative bacteria cells is that it has an outer membrane. Okay, so all cells have a plasma membrane, and then it's got this peptidoglycan layer, and outside of that is another phospholipid membrane. Okay, so that's a phospholipid bilayer. Much like the plasma membrane, which we haven't talked about yet. And within that outer membrane, there are porin proteins. Those are just for transport through that membrane. And also a structure called lipopolysaccharide. All right, we usually abbreviate that LPS. And that's composed of two polysaccharides and lipid A, which is also known as, AKA also known as endotoxin. And that is important because whenever the cell wall is disrupted, then that's released. So the cell wall, it's going to be disrupted when the cell dies to a huge extent. But really, anytime the cell divides, that has the potential to release a little bit of that lipid A. And that can cause some inflammation if it's an organism that's within a host cell. Okay, so when the cell wall is disrupted, that lipid A is released and can cause inflammation. We're going to talk more about the effects of endotoxin later, but I just wanted to throw that at you because it's a little bit confusing. It's the same thing as lipid A. Lipid A is endotoxin. All right, so if we label the parts of this gram-negative diagram here, down here we've got the plasma membrane. This pink part here, that's peptidoglycan. And then here's the outer membrane. Over here to the right, this is a porin protein. They kind of look like a couple of paper towel rolls there. Those are just for transport in through that outer membrane. There's some other proteins embedded in there. Probably most of them are maybe transport proteins as well. And then these hair-like structures on the very outside of the cell wall, that's the lipopolysaccharide, the LPS. Okay, and I want to mention that whether the cell is gram-positive or gram-negative, that doesn't dictate the shape of the cell. Okay, so there are gram-positive cells that are bacilli shaped. There are gram-negative cells that are bacilli shaped. There's gram-positive cocci, there's gram-negative cocci. Sometimes, because we only see certain things in lab, that students get that misconception. All right, here is a blow up of that lipopolysaccharide. This is from your textbook. Here's the lipid A that's embedded in the cell wall, okay, otherwise known as endotoxin. And then these two polysaccharides sticking out from that. You see here that this is labeled O antigen. That might sound familiar. There was a strain of bacteria that made the news kind of at the end of the summer, E. coli O157H7. It's a really nasty strain, not the strain that lives in our colon and helps us digest food. That's not dangerous, but this is a strain that causes really severe food poisoning, has the potential to damage kidneys. I think a few people died from it. I forget the cause of that, the source of question.
So this whole thing is a lipopolysaccharide.
Yes. So this is a blow up of these little hairs here that we're seeing in this picture. But that O antigen, this strain E. coli O157H7, this is the 157th version of that O antigen that they found. So there's variations of that O antigen, and that gives the even though it's all the same species, E. coli, there's lots of different strains, lots of kind of variations of the species. So that's where the O157 and the H7 is the 7th variation of the protein flagellin, which makes up the bacteria flagella. So just a little aside there. All right, so I like to show the cell wall different ways to draw it, just because it kind of helps us notice different things. So here's the drawing from your OpenStax textbook. You see the gram-positive here on the left, draws the peptidoglycan as these blue rectangles, and it's got teichoic acid. Then the gram-negative is a lot more complicated. Don't worry about this murine lipoprotein. We didn't talk about that, but it shows the LPS here, kind of labels the parts of it. Oh yeah, so lipopolysaccharide with the O antigen and the lipid A as part of the outer membrane. Okay. There is one other kind of cell wall that I want to discuss, and that's called the acid-fast cell wall. And this isn't nearly as common. Most bacteria are considered gram-positive or gram-negative. Your textbook actually considers this cell wall as gram-positive. So I'll just make a note of that. OpenStax considers this gram-positive. I've been taught to call this kind of a special kind of cell wall distinct from gram-positive or gram-negative, because cells that have this kind of cell wall don't take up the gram-staining dye very well. So we don't really often consider them one or the other. But again, OpenStax considers them gram-positive because there's no outer membrane like the gram-negatives have. So either way, we'll keep a little open mind. But what's special about these, they still have peptidoglycan, so don't forget that. But they have another major constituent, mycolic acid. And this is a long chain fatty acid. Okay, so when you see fatty acid, you think, okay, this is a cell wall that's high in lipids. So that gives the cell wall some really unique properties, kind of, if you think kind of waxy, you think wax paper. That's kind of what is encompassing these cells. So that, again, gives these cells some unique properties. they are very resistant to drying out. Now, we said that that was one of the functions of the capsule. That's why so many bacteria cells produce some kind of a glycocalyx or capsule. But an acid-fast cell wall, if that organism, say, is coughed out on your lungs and lands on a desk or something, that can stay viable for weeks if it has an acid-fast cell wall, where maybe a capsule might protect a cell for a day or two. Okay, so really super resistant to drying out, really quite resistant to chemical disinfectants as well. Okay, so this makes the cell very hardy, very environmentally stable, in other words. One drawback could be that the cells grow slowly. It just takes them a long time to manufacture these long chain fatty acids. So that's maybe not a problem for survival for these organisms. It certainly makes them more difficult to study because sometimes it takes weeks and weeks for colonies to grow up, okay, rather than, say, the overnight incubation that we're used to with a lot of other organisms. So they grow very slowly. The infections that they cause take a long time. They're kind of, chronic. They take a long time to actually kill someone. But there are a couple of famous infections caused by acid-fast organisms, primarily in the genus Mycobacterium. There are a couple of other genera that are considered kind of partially acid-fast, but this is kind of the main one that we think of, the Mycobacterium. And infections responsible, organisms responsible for the infections, leprosy, and the big one is tuberculosis, are caused by Mycobacterium organisms. So that Mycobacterium tuberculosis, that organism has been called the most successful pathogen of mankind. So that's one that has been a huge scourge that we will spend some time talking about more at the end of the semester. So if you're in lab, you'll get a chance to stain these organisms using this special acid-fast staining procedure. We have to use some different dyes because of that lipidy cell wall. Our kind of normal dyes that we use when we're gram staining don't penetrate that lipid cell wall. So we have a whole nother staining procedure. And if you're in lab, hopefully that will look something like that when you do it next week, not next week, but the week after. All right, so here's just the picture, another picture of acid-fast staining there from OpenStax, and kind of there's some other things here in the cell wall that we're not going to worry about, but here you can see the mycolic acid. Still lots of peptidoglycan, like any bacterial cell. Looks like there's a little teichoic acid in there too. So perhaps that's another reason that it's considered gram-positive, or more likely to be considered gram-positive. Okay, so as we finish the cell wall here, one more comparison slide of the three different kinds of cell walls. So again, I like to look at things lots of different ways. I want to think just for a minute before we move on. And I tried not to give this away as we went, but if you had to think about infections caused by gram-negative organisms, by gram-positive, or acid-fast, what do you think? Which one do you think is going to be most difficult to treat with antibiotics? Okay, so maybe think about that. Talk to the person next to you and see if you can justify your thought process. So I'll give you a minute to do that. Okay, so what are you thinking? If anybody want to hazard a guess, what's going to be most difficult to treat? Okay, what do you think? Okay, what you thought, Passos? Why mycobacteria?
Says resistant chemical disinfectants. You said tuberculosis is known as the world's most successful pathogen.
Okay, right. So yes, you're right. It's the mycobacterium that are most difficult because the antibiotics don't penetrate through that lipid cell wall. Some of you may have thought gram-positives are harder to treat because they have all this peptidoglycan. But the peptidoglycan, basically, if something's small enough to fit through there, it's just kind of holes through there, and there's no problem with the penetration. Yeah.
Do you have a diagram for the mycobacteria like you did for the other two?
Do I have a three-dimensional diagram? I have not found one like that. So no, that would probably be quite complicated because there's other stuff in the area that we didn't really talk about too. Okay, so yes, tuberculosis, mycobacterium, difficult to treat. Second most difficult is gram-negative. We have less antibiotics that are going to be helpful against gram-negatives, again, because of that permeability issue, because of that outer membrane. It's just kind of another barrier for the antibiotics to have to penetrate through to actually get into the cell to do some good. Okay, and then we have more choices for the gram-positive. It's good. So let's continue working into the cell. I'm not going to spend much time on the cytoplasmic membrane because you studied this in other classes before, I'm assuming, and prokaryotic membranes are really similar to our membranes. That's one of the reasons that we are not very successful in our antibiotics that target prokaryotic cell membranes are pretty toxic to us as well, because they're very similar. But of course, the function, it's a semi-permeable barrier, meaning that it lets some things through and some things not. Also, there's a lot of energy reactions that we see are going to, glycolysis takes place in the cell membrane. It's really important that that membrane is intact. Otherwise, the cell can't kind of maintain the gradients that it needs to in order to produce energy. All right, so again, lipid bilayer. Within that membrane, there has to be ways for the cell to transport things in and out. So if we're talking about transport, any passive processes, that means that there's no ATP required. So things like diffusion, that's just particles that are moving from a high concentration to a low concentration. We call that down the concentration gradient. So there's some things that can diffuse through a cell membrane without any special protein channel, things like water, of course, diffusion of water, we call osmosis. Things that are really small, things like simple sugars, glucose, gases, some ions, anything that's lipid soluble perhaps can pass through without a protein to kind of shuttle it through. If there is some sort of transport protein necessary, we call that facilitated diffusion. And sometimes a protein channel will work for lots of things to passively pass through. Sometimes those channels are more specific, and it might be a special permease enzyme that only recognizes a certain thing to pass through. I want to spend a little time on osmosis just because you've had this before. I find there's still confusion about it, and it's one of those big themes in biology that you just need to understand a lot of other things. So these terms, isotonic, hypertonic, hypotonic, remember those are relative terms. So you would never say that solution is hypertonic. Well, compared to what? Okay, you always have to compare it to something. And I think the thing that trips people up sometimes is that we use those terms to describe the medium that the cell is in. It's not correct to say the cell is hypertonic. And the reason is because the chemistry of the medium can change quite rapidly, where the cell chemistry is not likely to change very, very rapidly. So we use those terms to, again, refer to the medium relative to the cell. All right, so if the medium is hypertonic, that means that there's going to be a lot more solutes, something that can't pass through that semi-permeable membrane of the cell, something like salt, for instance. There's more solutes outside of the cell than in. And so essentially the water moves to try to equilibrate that concentration. A student said to me once, water follows stuff. All right, so if there's more stuff outside of the cell, the water is going to move out of the cell. All right, so this is the really bad situation for bacteria cells, because the cells are going to, or any cell is going to dehydrate. All right, anytime they're losing too much water, then that can't support, the cell can't be viable. Of course, in a hypotonic solution, that means there's a lower amount of solutes outside of the cell than what there is in. And so water is going to be rushing into the cell to try to equilibrate that. Did I say it the wrong way? Do you have a question? Do you have a question? Okay. Oh, why do your fingers get wrinkly? I don't know. That's a question for your physiology professor. Yeah, I don't really know. I have to think about that. But so hypotonic medium, water is rushing into the cell. And if this was a red blood cell or maybe some kind of animal cell, it wouldn't take very much of a concentration gradient for that to burst. But since we're talking about bacterial cells, which you know has a nice, most of them have cell walls, they don't actually mind a little influx of water, slight hypotonic medium, because that cell wall keeps them from bursting the same way that a plant cell wall likes a little bit of turgor pressure. Okay, so it takes a huge concentration gradient for that bacterial cell typically to burst, as long as it's intact. If it's been weakened a little bit, say by some antibiotic, then that's when it's going to burst. All right, so make sure you have straight kind of which way the water moves in what kind of situation there, because we're going to come back to that a few times this semester. Okay, and again, continuing with transport, any process that's considered active, that means that ATP is required to transport, you know, whatever, if we're talking about a nutrient, for example. And I'm not too caught up in these terms, uniports, antiports, coupled transport. Basically, ATP is required, and sometimes it's energetically more favorable to, for instance, maybe let one ion diffuse out, and that will help something else be, you know, less energy to come in. But again, active ATP is required. One example of an active process that is unique to prokaryotes is called group translocation. And this is actually, the best example of this is the first step in glycolysis, where glucose is phosphorylated. But in group translocation, basically there's a special protein that's going to modify that substance that it's specific for as it enters the cell. So in this case, glucose is phosphorylated and becomes glucose 6-phosphate. Okay, so the big advantage here is that now you've changed glucose if there happens to be a lot more glucoses inside of the cell than out, we don't want that glucose to diffuse right back out again, or the bacterial cell doesn't want that. So if you change it, now it's got a phosphate on it, and now it's not going to fit back out through the membrane. So we've modified it so we can't lose it again. Okay. All right, so just kind of where we're at, again, we're working into the cell. We've gone through the capsule, the cell wall, cell membrane, talked about flagella, any fimbriae, or any appendage like that. We're ready to finally move into bacterial cell. And there's not a whole lot in there. So don't worry that this is going to take forever. There's cytoplasm in there. Obviously, that's the just kind of whatever you find inside the cell membrane. Typically, very often you see those terms cytoplasm and cytosol kind of used interchangeably. Technically, the cytosol is the fluid part of the cytoplasm. I'm not really interested in using that quite correctly all the time, but there's proteins floating around in there, dissolved ions and things like that. And within the cytoplasm, there is a nucleoid region. So where the DNA of the cell is, remember, there's not a nucleus, but it is concentrated in this nucleoid region of the cytoplasm. Again, there's not a membrane around that. So within that nucleoid region, we're going to find the chromosomes of the bacterial cell. And I put an S on there, but usually the bacterial cell is either going to have one or two. Okay, so not a huge number of chromosomes, like what a lot of eukaryotes have, and they are usually going to be circular, okay, rather than linear. Okay, a circular chromosome is just a lot more stable. Anytime you have a linear chromosome, those ends are subject to being degraded by endonucleases, so a lot more stable. Also, a bacterial cell will have plasmids, will have some DNA in the form of plasmids. These are circular, and they are much smaller than the chromosomes. It may have quite a lot of them. We'll talk a little bit more about the form that the DNA takes in bacterial cells when we get to our genetics chapter, but typically a plasmid is not going to encode for anything that's really integral to the survival of the cell. You're not going to have things like directions to make enzymes for glycolysis. You're going to have things like that might help it survive in certain circumstances. So you might have antibiotic resistance genes on a plasmid, or something that may help it cause an infection. Okay, so those are just examples. Okay. Another big structure that any cell is going to have is a ribosome. So, and of course, the ribosomes are very important because they're the site of protein synthesis. So we'll talk much more about protein synthesis later. But what you should know about the ribosomes is that there are actually a small and a large subunit that come together to form the ribosome complex. So we talked about how prokaryotic cells don't have membrane-bound organelles. This is not a membrane-bound organelle. If you had membranes around each of those subunits, and then when they came together around the messenger RNA to make the proteins, that just wouldn't work out. So they're not surrounded in a membrane. Question? Yes, yes. So we'll get to that. I'm going to go ahead and compare the prokaryotic and the eukaryotic form right at the same time here. What are the ribosomes made of? They are made of ribosomal RNA and proteins. Okay, so, right, you guessed it here. There's two different forms. So the prokaryotic form of ribosomes and a eukaryotic form. Major difference in size. So the prokaryotic form, we have this 50S subunit, that's the large subunit, and a 30S small subunit that come together to form a ribosomal complex that's considered 70S. Okay, what does that S stand for? The S stands for Svedberg's, which is a unit that's based on sedimentation. A unit that measures the sedimentation rate. Okay, so it's kind of confusing. How can you have a 50S and a 30S come together to form 70S? That doesn't add up. That's because, again, we're measuring sedimentation rate, which is kind of a function of the weight, the shape, the overall kind of size of a structure. So it's not linear in terms of the measurements there. So if you're spinning ribosomes in a centrifuge and you have them in some kind of viscous material, like how quickly they sediment down to the bottom, it's a measure of that. Okay, and of course eukaryotes here, it's just a little bit different size. The 60S and 40S measures that 80S ribosomes. Okay, so this 16S subunit of ribosomal RNA, that's the one that Wose and Fox sequenced when they were trying to put organisms into groups. Okay. Other structures that may be present. Some bacteria have inclusions, which are probably the closest thing that they'll come to is having organelles, because they're not really enclosed in a membrane, but sometimes there's maybe a little proteinaceous film that will kind of keep them, all these inclusions all together. And what are inclusions? They are storage sites. So typically, there's a wide variety of different inclusions that cells may have, typically storing something that they may be able to, again, accumulate in times when it's plentiful, and then in times when it's not available, they can use those storage sites. So things like lipid, starch, phosphates, it could be a lot of different things. So just some examples of inclusions. Probably the coolest one here is that some organisms can store up polyhydroxybutyrate. And that is a starchy polymer that some cells can accumulate, especially if you grow the organisms in certain medias. I think I forget what kind of organism that is in that picture, but if you grow it in a medium without nitrogen, then it forms lots and lots of these PHB inclusions. And the cool thing about that is that we have learned how to make cells do that on purpose, and we can make biodegradable plastic out of that PHB that the bacteria cells make. I just love examples when we can use bacteria to do stuff for us that makes our life better. Because obviously the plastic that we make out of oil is basically stays in landfills forever. So PHB plastic, I went back and looked at this website, and it doesn't really say very much about the process, but I think over a period of a few months, it will biodegrade. So this, an application for everything. Some kind of plastics you don't want to break down that quickly, but there's a lot of medical applications. It is more expensive to make plastics this way, but it's kind of a exciting thing that, you know, something may come of it eventually. Other examples of inclusions, volutin, some organisms can store inorganic phosphate. That's something, a lot of phosphate is necessary to during metabolism. And sometimes that's also called metachromatic granules, because you can see that in this picture here. Here's an example of volutin granules. Sometimes that shows up when you're staining cells. So again, certain kinds of cells. Magnetosomes, that's when cells store some form of iron, whether it's iron oxide or iron sulfides, either way. But we think that some cells can use that to orient themselves. So it's kind of a magnetic compass type thing to aid their movement. And simply sometimes cells can store up gas in order to orient themselves in the water column in the right space if they're photosynthetic bacteria, for example. Okay, so those are just some examples, and your figure in your book here shows some pictures that kind of show some of those. This first one is PHB. Biotin, yeah, here's some magnetosomes there. So again, very specific. Some organisms might not make any inclusions at all. Okay, and really this is the last thing that we want to talk about in the inside of the bacterial cell, is that some bacteria can form endospores. And endospores are not reproductive. It's kind of confusing because when we get to talking about fungi produce many, like hundreds of spores, which eventually grow into a new fungal organism. Endospores are not reproductive for bacteria. It is more of a survival mechanism. Okay, they kind of hunker down into this kind of thick-walled endospore if conditions suddenly become not optimal. Not very many bacteria can do this. The most famous ones that can are in the genus Clostridium or Bacillus. There are a few others. Some of those other genera used to be considered a Bacillus or a Clostridium. These are a couple of genera that are gram-positive rods. And these are organisms that are usually found as vegetative cells. So that means that they're metabolically active. And when they're in their vegetative state, they're really susceptible to all the things any normal bacteria cell would be susceptible to. But when they encounter some kind of harsh environment, they can actually sporulate into this endospore state. So what might be some examples of harsh environments that a bacteria might encounter? Okay, yeah. All right, extreme temperature, especially hot temps. They can survive cold ones pretty good, but especially hot temperatures. What else? Yeah. High salinity, maybe. Okay, yes. Okay, so high salt. What else would be the harsh environment? What about, yeah. Dry, yes. We're just saying they don't like dry conditions. All right, you can probably think of lots more, yeah? Well, if it was sterilized, it probably killed them. Yeah, okay, you're on the right track. We could say low nutrients. We could say exposure to radiation, like UV lights or gamma rays, something like that. pH fluctuations, like, okay, that's, most bacteria like warm, moist conditions. And anything other than that could potentially not be ideal. We'll talk about some of those physical conditions later. But we're out of time. I think we'll be able to finish out chapter 3 on Monday. So have a good weekend.